Good morning, dear students. This is Dr. Sjctnc-Pravina Mary, Department of Zoology. Today we are going to learn about recombination DNA technology or rDNA technology. The recombination DNA technology is otherwise called a gene cloning. It is a novel technique for controlled recombinations. Recombination DNA technology involves using enzyme and various laboratory techniques to manipulate and isolate DNA segments of interest. It is a method of bringing together and joining pieces of DNA from unrelated organisms. Its example bacteria and the human, achieving the aim and profit of general recombinations. These techniques mainly involve in vitro genetic manipulations. Next we are going to learn discovery of recombination DNA technology. The DNA structure was first described by Watson and Crick in the year 1953. The isolation of DNA ligase in the year 1967. rDNA technology developed by Paul Berg, Cohens and Bayer in 1973. First plasmid vector capable of being replicated with bacterial host. So next we are going to learn goals of rDNA technology. So we are following some goals like to isolate and characterize genes, to make desired alteration in one or more isolated gene, to return altered gene to living cells, artificial synthesize new genes, improve human genome, understand hereditary disease and their cure, alterations the genome of an organisms. Next we are going to learn steps of rDNA technology. In these techniques, a particular desired gene is isolated from one organism and introduced to another organism using a suitable vector. The insert desired genes are replicated independently and is transferred to progenesis as a result of cell divisions. The genetic engineering techniques involved 5 main steps. They are preparations of desired gene, isolations of plasmid vector, insertions of desired gene into vector, introductions of rDNA in host cell, and last one identifications of gene cloning. So today next we are going to learn isolations of desired piece of DNA. So desired gene can be obtained in 2 ways. One is a natural source, another one is a mRNA. So first we are going to learn natural source. The desired gene, say insulin gene, is available in mammalian pancreas. The desired gene is isolated from the cells by following steps. The first one, the cells are washed with a sodium chloride solutions, and then cell wall rupture by homogeneations. The homogeneations is treated with the detergents like sodium sulfate. The homogeneate is centrifuged in removed cell debris. The supernatant contains DNA, RNA and protein components. The proteins are precipitated and proteinase and removed by these centrifugations. The supernatant is treated with ribonuclease. Its centrifuge is remove RNA. The solutions is treated with ethanol and stirred well with a glass rod until a white fibrous precipitate is formed around a rod. The precipitate is dissolved in ethanol sodium chloride solutions. It contains DNA. So next we are going to learn isolations of plasmids. So plasmid means it's a circular, double-stranded, self-replicated component. It's found in a bacteria cells. The plasmids is isolated from the bacteria cells. We know it. So the following steps we have followed. The bacteria cell wall is ruptured by treated with lysoenzyme and the ETTA. The cell is lysed with a treatment with sodium sarsenate. The plasmid DNA is separated from other cell component by centrifugations method. So next we are going to learn insertions of desired gene into the plasmids. So after the preparations of desired gene and plasmids, the desired gene is inserted into the plasmid. It involves the following steps. The first one, plasmid is cut with restriction enzymes. Restriction enzymes, it's found as chemical knives in genetic engineering. The next one, restriction enzymes make two types of cuts. One is the EcoR1, make cut producing DNA fragment with sticky ends. Then second one, the desired gene is also cut with the same restriction enzyme. The enzymes producing DNA fragment with sticky ends or blunt ends. The cut enzymes and desired gene are mixed together. The desired genes get inserted to the plasmids. The sticky ends of plasmids and desired genes are linked by complementary base pairing. This process is known as an ligation. So next we are going to learn introductions of rDNA into host cell. The recombination DNA can be introduced by a bacterium, plant cells or animal cells. The cell that receiving the foreign DNA is called transformed cell, and this process known as transformations. So transformations is a process of genetic modifications in which fragmentations of DNA from one cell enter and get integrated to the DNA of another cells. Or DNA introduced into the host cell by any one of the following method. One is the transformations, microinjection, electroporation, shotgun method, and liposomes mediations or fusions. So first we are going to learn transformations. So transformation means rDNA can be introduced into bacterial cell, that is known as transformations. Transformations is a genetic modification process where DNA fragment of one cell enter and get integrated into the DNA of another cell. Our DNA is mixed with a culture of E. coli. The E. coli take up rDNA from the medium and is integrated with a genome of E. coli. This E. coli is transformed. The next we are going to learn electroporations. So electroporation, it can be introduced by plant cells by electroporations. The electroporations is the introductions of rDNA into plant cells through cell wall by making the cell wall permeable with electrical shock treatments. So next one is the shotgun method. Shotgun method, rDNA can be introduced into plant cell. rDNA coated a golden particles are injected into plant cell by a micro projecting gun. Then fourth one is the micro injections. Micro injection means it's introduced into a plant cell, animal cell and egg by syringe. This method is known as the micro injections. So then last one, identifications of gene cloning. The host cell containing rDNA is called transformed. The process of rDNA is a cloned gene is the host can be identified by the following method: insertional inactivations, immunochemical method, and cloning hybridizations. So first we are going to learn insertional inactivations. The inactivations of antibiotic resistant can in a plasmids by the insertions of desired gene is known as insertional activations. The next one is the immunochemical methods. When the desired gene produced an antigenic protein, the rDNA can be identified by immunochemical method. The protein produced by the recombination cells are allowed to bind with a radio-labeled antibiotics. The clone which binds with a radio-labeled antibiotics contains rDNA. The last one, clone hybridizations. Clone hybridizations in this method, the rDNA can be identified by mixing with a radio-labeled complementary mRNA. The rDNA of transformed cell along with a base pair with mRNA. Next we are going to learn Goals of rDNA technology: to isolate and character of genes and artificial synthesized, and the understanding hereditary disease and their cure, improve human genome. That is the main goal of rDNA technology. Thank you, students.